Hello friends, welcome to SAM Biology class. Friends, today we shall discuss about the mechanism of crossing over. Before that, I request all of my friends, those have not subscribed my YouTube channel, please subscribe. Also watch, share and like my videos. Crossing over is the reciprocal exchange of genetic material between the non-sister chromatids of homologous chromosomes. Chromosomes which have same genes in the same order along their arms are known as homologous chromosome. Because of this exchange of genetic material, new combinations of genes arise. So the result of crossing over is genetic recombination. The mechanism of crossing over involves different steps such as synapsis, duplication, crossing over and terminalization. Step 1 is synapsis. Synapsis is the close pairing between homologous chromosomes. It occurs during the zygotine substage of meiotic prophase 1. In this figure, these are two homologous chromosomes. Each homologous chromosome consists of two sister chromatids. Crossing over always takes place between non-sister chromatids. The chromatids of these two homologous chromosomes are related as non-sister chromatids. But the sister chromatids never exchange genetic material during crossing over. In zygotine, homologous chromosomes come closer and start pairing. Pairing is initiated in the centromeric region, then it proceeds toward both the ends. Pairing between homologous chromosomes is precise and point to point. The force of attraction that exists between the non-sister chromatids contribute to this pairing. But the question is, why do homologous chromosomes pair? To answer this, several hypotheses have been proposed. One of the hypotheses is precocity theory, which was proposed by C.T. Darlington in 1937. According to this theory, chromosomes entering the meiotic prophase are unreplicated structure consisting of single chromatids. Such unreplicated chromosomes represent an unsaturated and unbalanced state. To become saturated, homologous chromosomes pair up. As soon as the chromosomes duplicate themselves, in the Pakitian stage of meiosis, they start separating. Another concept is proposed by Stern and others. According to this concept, shortage of DNA and histone during zygotine create a favorable condition for this pairing. Various evidences indicate about the formation of a complex between homologous chromosomes during synapsis. This complex, which is a highly organized structure of elements, is known as synaptonemal complex. This complex has been described by Montrose J. Moses in 1955. Synaptonemal complex is formed between homologous chromosomes during synapsis. This highly organized structure consists of a central element and two lateral elements. The lateral elements are flanked on both sides by chromatin of sister chromatids. The central element is of variable prominence, but the lateral elements are very dense. At the center, a nodular structure is seen, which is known as recombination nodules. The electron micrograph of the complex also show the presence of fine transport strands which pass from one lateral element to other through the central element. The protein which is present in this complex is known as synaptonemal complex material. It is because of close pairing of homologous chromosomes during synapsis, exchange of genetic material becomes possible. So synapses provide the mechanical basis for genetic variation. Synapsis is followed by duplication, which occur during the Parkinson substage. In this step, the paired homologous chromosome undergo longitudinal splitting, thereby separating the two sister chromatids which still remain attached at the centromere. At this stage, the paired homologous chromosome, which consists of 4 chromatids, is known as tetrad. Third step is crossing over, which takes place during diplotin substage. During diplotin, as the paired chromosomes start separating, the non-sister chromatids remain in contact at one or more points, thereby establishing one or more exchanges of genetic material between them. The point at which the non-sister chromatid remain in contact is known as chiasma. At these points, the non-sister chromatids undergo breakage and rejoining. As a result, exchange of genetic material takes place between them. According to Stern and others, breakage of non-sister chromatid is mediated by nuclear endonuclease and rejoining by ligase. Last step of crossing over is terminalization, which occurs during dikinesis. After crossing over, the non-sister chromatids start repelling each other. The separation of non-sister chromatids start at the centromere, then proceed toward both the ends. In doing so, the chiasma also move in a zipper-like fashion. This movement of chiasma toward the chromosomal ends is known as terminalization. Simultaneously, the chromatids condense and shorten up, and during dikinesis, the homologous chromosomes are separated from each other except at the chromosomal ends. It is because of breakage and rejoining of non-sister chromatids and chiasmata, reciprocal exchange of genetic material takes place between homologous chromosomes. As a result, new combinations of genes arise. Because of this exchange of genetic materials, the resulting chromatids contain the genetic material of both of them. Such type of chromatids which are formed as a result of this genetic exchange are called as recombinant chromatids, whereas the remaining chromatids which do not participate in crossing over retain their original genetic constitution. Such chromatids are called as non-recombinant chromatids. So friends, the process of crossing over gives rise to genetic variability among population. So friends, this is all about the mechanism of crossing over. For more updates, please subscribe my YouTube channel, Sam Biology Class.